1, this question is about the decay of milk. Name 2 types of microorganism that cause decay. Well, we've got bacteria and fungi. Cow's milk is pH 6.6. As milk decays, lipids in milk are broken down. One of the products of the breakdown of lipids causes the pH of milk to decrease. Name the product that causes the pH to decrease. Well, if you remember, lipids are made up of fatty acids and glycerol. So if we're looking for a pH decrease, then it's becoming more acidic, so it's going to be the fatty acids. A student investigated the effect of temperature on the time taken for different types of milk to decay. This is the method used. Put cow's milk in 6 test tubes, keep each test tube at a different temperature, measure the pH of the milk in each tube every day for 12 days, record the number of days taken to reach pH 5, and then repeat the steps 1 to 4 with goat's milk and with almond milk. Give one way the pH can be measured. The most obvious piece of equipment to use would be a pH meter, but you could also use universal indicator paper or solution. Give 2 control variables the student should have used in this investigation. Well, if we go back and have a look at the method, we can see that the independent variable is the effect of temperature. So that's not going to be a control variable because we are going to be changing that temperature. And it says put cow's milk in 6 test tubes, but it doesn't actually say how much. So that's a control variable we can write down in a minute. Keep each test tube at different temperature. Well, that's our independent variable that we've already spoken about. Measure the pH of the milk in the tube every day for 12 days. So really we should do that at the same time of day. So for our 2 control variables, let's put the volume of milk and the time of day the pH was measured. The student improved the investigation to produce valid results. Figure 1 shows the results. So we've got a graph and it's showing the time taken to reach pH 5 in days and different temperatures. Now we've got different types of milk. We've got our key there, cow's milk, goat's milk and almond milk. Which type of milk stays fresh the longest at 10 degrees C? So let's look on the graph at 10 degrees C. We're looking for the milk that takes longest to reach pH 5, so which is the highest one there. So we've got almond milk. Describe the effect of temperature on the time taken for goat's milk to reach pH 5. Use the data from figure 1 in your answer. Well, as the temperature increases up to 15 degrees C, the time taken to reach pH 5 decreases. And then if you have a look at the graph, you can see as temperature increases above 15 degrees C, the time taken to reach pH 5 actually stays the same. The time taken for cow's milk to reach pH 5 at 10 degrees C is less than the time taken for cow's milk to reach pH 5 at 5 degrees C. Suggest one reason why. Well, they're both pretty cold temperatures, but the enzyme activity is definitely going to be faster at 10 degrees C compared to the 5 degrees C because there will be more kinetic energy at 10 degrees C. So that means the bacterial enzymes can break down the lipids and convert those into fatty acids and glycerol much more quickly. Suggest 2 reasons why the different types of milk took different lengths of time to reach pH 5. So it's a suggest question. That means we need to just take an educated guess. So let's go for different milks could have different concentrations of fats or lipids. Maybe the different milks had a different starting pH. The student said the temperature milk is stored at affects how likely the milk is to cause food poisoning. How can the investigation be developed to find out if the student is correct? Determine the types of bacteria present in the milk. Record the pH every 12 hours. Use more than 3 different types of milk. So it's going to be determine the types of bacteria present in the milk. Not all bacteria will cause food poisoning. So we need to just find the ones that do. Number 2, figure 2 shows the human population from 1600 to 2010. So we've got the year on the x-axis and then the human population in billions on the y-axis. In 1900, the human population was 1.6 billion. Calculate how many times greater the human population was in the year 2000 compared with the year 1900. So if we go to the year 2000, then we can see that there were 6 billion people in the year 2000, and compare that with 1.6 billion, or how many times of 1.6 go into 6. So the human population was 3.75 times greater in 2000 than it was in 1900. In 1950, the human population was 2.5 billion. Calculate the mean annual increase in the human population between 1900 and 1950. Well, we already know that there were 1.6 billion people around in 1900. We need to do 2.5 billion minus 1.6, and we need to divide that over 50 years because that's the time difference. And that will give us an annual increase of 0.018 billion per year. Predict the human population in 2050. If the current rate of population increase continues, you should draw an extrapolation line on figure 2. So we need to go to the graph and we need to carry that line on from where it looks like it's going to go. And when it hits 2050, it actually looks as if there will be 10 billion people if it carries on how it is. The increase in human population has caused a decline in fish stocks. Describe how fishing quotas can help to return fish stocks to a sustainable level. If you catch fewer fish, then that means the remaining fish will be able to reproduce. Farming techniques have changed in recent years. Describe why more land is being used for farming and how increased farming has decreased biodiversity. An increase in human population leads to a demand for more food. Therefore, more crops are needed. Crops consist of plants that are the same type though. So this means there'll be very little biodiversity of the plants and therefore less biodiversity of the animals in the area as well. Creating areas to grow crops or keep livestock also means that the plants that were already in the area have to be removed. Vast numbers of trees will be removed to create space, and this will lead to deforestation, causing a dramatic reduction in biodiversity of plants and animals and fungi and bacteria and all sorts. Loss of biodiversity there and loss of habitats too. Farmers will use herbicides and pesticides on their crops, and that could disrupt food chains, reducing the biodiversity. They will also use fertilizers, and we know the problem when we have fertilizers is that when it rains, it can wash into nearby water, and then that can lead to water pollution and the problem of eutrophication, which will reduce the biodiversity in the nearby water, such as lakes. Okay, this question isn't really about the details of eutrophication because it's 6 marks about something else, but eutrophication is when the algae grows over the top of the lakes and it blocks the light from getting to the plants at the bottom of these lakes. They can't produce oxygen for all the organisms living in that water, so there's less oxygen in there. And then even worse, then they die and the decay and then the increase of bacteria in there. They use the oxygen for their microorganism respiration, so they're taking the oxygen out of the water as well. So we're going to get a big impact on the animals and plants that should be living in that water body usually. Also, rearing livestock, cattle, requires vast areas to be cleared, and the increase in numbers of the cattle also leads to an increase in methane production, which is a greenhouse gas, so it's going to contribute to air pollution and the greenhouse gases, therefore global warming. Genetic modification of crop plants can help meet the demands of the increasing human population. Golden rice is a genetically modified crop What is the advantage of golden rice compared with non-genetically modified rice? Tick one box. Golden rice contains protein-rich mycoprotein. No, it doesn't. Mycoprotein is that protein made from fungus. It's not that. Golden rice has improved nutritional value. Yes, absolutely. Golden rice produces human insulin. No. Just checking that third one. It's definitely the middle one. Suggest one reason why some people are concerned about the use of golden rice. There are lots of reasons people get worried about these things. So the main one I would put down there is that people worry that the gene might enter into other species. Maybe we will get this reduction of our actual traditional wild rice, and therefore the insects that like to live around those areas, feed in those areas, we're going to get a reduction of the biodiversity around our traditional wild rice. This question is about plant hormones. Farmers can spray seeds with gibberellins to start germination. What are the two other uses of gibberellins? Okay, it's asked you to tick two boxes, so make sure you only tick two. Is it going to help in tissue culture? No. To help roots to form? To increase fruit size? Yes. To kill weeds? No. To promote flower production? Yes. Students investigated the effect of light intensity on the height of seedlings. This is one of our required practicals. Figure 3 shows the equipment. Describe two improvements the students should make to their investigation. Can you see here that there's a radiator one end and not the other end? So we need to make sure that we keep the temperature the same for all the seedlings. And you might notice also that there's an uneven number of seedlings in each of these Petri dishes. So we do need to make sure there are equal numbers of seedlings in each dish. There are other things also that you might put, but obviously you're only going to describe two. Make sure there is just one on each line because the examiner is only going to look at the first one on each line. Other things that you might have thought about is use seedlings of the same height. That's going to be really difficult to do that, but you could write it, I suppose. Use more seedlings in each dish, but make sure they're the same number. Give all of the seedlings the same volume of water. Use the seedlings of the same species, and so on. Figure 4 shows a seedling growing towards a lamp. Suggest how the students measured the length of the curved seedling in figure 4. Well, the easiest thing I think I would put there would be to measure it with a flexible ruler or a tape measure. But you could also use a piece of string. So get a piece of string, line it up along the seedling, and then just mark on the string the start and the finish. Pop it against your ruler, and then you can use that to measure it. Explain what happened to the growth of the seedling on side Q compared with the growth on side P. So let's look back at our diagram. We can see that on side Q, which is away from the light, that more elongation has happened there because we know that auxins move to the shady side. And on side P, we've got less elongation of cells, so less growth there. So side P receives more light than side Q. The auxin moves to the shady side, which is Q, of the plant, causing an unequal distribution. They do like you to say that causing an unequal distribution. The auxin causes the cells on the side Q to elongate, meaning that side Q is going to grow faster on the shady side. And because it's growing faster on the shady side, that's going to cause the seedling to bend towards the light. This is phototropism. Bananas are often stored separately from other fruits because bananas release a plant hormone. Why does storing bananas with other fruits cause the other fruits to ripen faster? That is because bananas actually naturally release ethene, which helps to ripen fruits. DNA is a polymer of nucleotides. Why is DNA described as a polymer? Well, DNA is made up of many monomers, and those monomers are called nucleotides. Figure 5 shows part of a DNA molecule. Describe the structure of a nucleotide. So we're just going to focus in on one area. We've got a sugar, a phosphate group, we've got a base, and these are our complementary bases. A phosphate group is attached to a sugar, which we can see clearly in the diagram, and the sugar is also attached to a base, and the base will be one of 4 possible bases. The length of a DNA double helix increases by 0.34 nanometers for every pair of nucleotides. The total number of nucleotides in a human body cell is 1.2 times 10 to the 10. Calculate the total length of a double helix in a human body cell. Give your answer in meters. So we've got some conversions we need to be aware of here. Now, with all maths, you know that there are different ways you can get to the same answer. So if you look at the mark scheme for this question, it might be slightly different to what I've done here, but it's the right processing and it gets you to the right point. Right, so how I would work this, if we've got a total number that's 1.2 times 10 to the 10, and I'll just pop all the zeros in there. If that's our total number, how many pairs have we got? So we just divide it by 2. So that means we've got 6 billion pairs of nucleotides. It said that if we go to the top of the question, 0.34 nanometers for every pair of nucleotides. So we've got 6 billion times 0.34 nanometers will give us actually how long that length will be in nanometers. Yep. But it wants the answers in meters, and because there are 1 billion nanometers in 1 meter, it just means we need to divide that by 1 billion. So we should end up with the total length being 2.04 meters. Some parts of DNA do not code for proteins. Describe how non-coding parts of DNA can affect the expression of genes. Non-coding parts of DNA can actually switch genes on or off. There are 2 types of cell division, mitosis and meiosis. Describe 3 differences between the processes of mitosis and meiosis. Well, mitosis produces 2 daughter cells and meiosis produces 4 daughter cells. Notice that I'm saying what mitosis does and what meiosis does. You can't just say mitosis produces 2 daughter cells and think that you're going to get a mark for it. You've got to make it a comparison. Same with this one here. So if we say that the daughter cells in mitosis are diploid, which means 2 sets of chromosomes, then we've got to say what the daughter cells in meiosis produce, and they are haploid. Mitosis produces genetically identical cells, and meiosis produces genetically different cells. Describe 1 similarity between the processes of mitosis and meiosis. I think this question should say cell cycle, but we'll go with it. The DNA has to replicate, and also don't forget, before we get this nuclear division, we've got to have an increase in the number of mitochondria as well as the DNA replicating. We've got to have increase in number of ribosomes, or you might get a mark for saying subcellular structures. I'm not quite sure how to say this word, so please bear with me here. Dupuytren's is a disorder that affects the hands. Figure 6 shows the inheritance of Dupuytren's in one family. We've got information that tells us it's caused by a dominant allele. So big D for dominant allele, little d for recessive allele. Before I even get to the question, what I really like to do is to put the genotypes up in the key. So if I've got a male with Dupuytren's, then they're going to have a dominant allele. So the genotype for that could be big D, or big D, little d. So that'll be the same for the female who has Dupuytren's. But a male without and a female without has to be little d, little d. Give the genotype of person 1, explain your answer. Now, number 2, who he has children with, has to be little d, little d. And we know that the offspring will get 1 allele from their father and 1 allele from their mother. So the fact that 5 and 6 don't have Dupuytren's means that they must have inherited a little d from their dad, person number 1. So therefore, dad has to be big D, little d. The genotype is big D, little d. It's a big D because they have Dupuytren's, but must have a little d because they have offspring that are little d, little d. They must have got 1 little d from person 1 and the other little d from person 2. Person 7 and person 8 in figure 6 are expecting a 4th child. What is the probability of the child having Dupuytren's? You should draw a Punnett square diagram, identify which offspring have Dupuytren's. So we need to know what the genotype of 7 or 8 are. We need to go back to the pedigree and we have a look. We definitely know person 8 doesn't have it, so they have to be little d, little d. And person 7 has to be big D, little d because they actually have offspring that don't have it as well. Pop them into a Punnett square. Person 7, big D, little d, these are the gametes, yeah. Person 8, little d, little d. Pop your offspring in there, big D, little d, little d, little d, big D, little d, little d, little d. And we can circle, show the examiner that we know which offspring will be affected. Those are the big D, little d's. They will have Dupuytren's, and there will be a 50% probability of the child having Dupuytren's. Explain how figure 6 shows the allele for Dupuytren's is not on the Y chromosome. Assigned females at birth are XX, assigned males are XY. Figure 6 shows that it's not on the Y chromosome because actually we have females who can get it and they don't have a Y chromosome. So if you have a look, you've got females 3, 11 and 12. They have Dupuytren's and females do not have a Y chromosome. Figure 7 shows the brain. Which part of the brain becomes more active if a person balances on 1 leg instead of standing on 2 legs? Should we have a quick whiz round labelling the brain because we can? A is the cerebellum, B the medulla oblongata, C the pituitary gland, and D the cerebrum or cerebral cortex. We know that the balancing is the cerebellum, so it's A. Name the part of the brain that is responsible for making a decision. That is the cerebrum or the cerebral cortex. In most MRI scanners, the person being scanned needs to stay completely still. A functional mRNA scanner allows a person to move while the scanner makes images of the person's brain activity. Suggest how the fMRI scanner could help to find out more about the brain damage a person has. You can ask a person to do a specific task to see which part of the brain is active or inactive. You can compare this to a person without brain damage to be able to identify where the actual damage is. Describe how the brain receives information about light entering the eye. You should include the names of structures in your answer. Photoreceptors in the retina detect light, and an impulse will travel along a sensory neuron, which are found in the optic nerve. The eyes of some birds contain cells that detect ultraviolet light. UV light is reflected by some fruits and the urine of small mammals. Explain how birds that detect UV light have evolved from birds that could not detect the UV light. A mutation in the DNA may have led to cells in the retina that can detect UV light. The birds with the mutation to be able to detect the UV light are able to see the fruits more clearly. And if they can see the fruits more clearly, they're more likely to get food, and therefore they're more likely to survive, reproduce, and pass on this beneficial allele or mutation to their offspring. And that is an example of natural selection. Also, this beneficial mutation or allele also enables the birds to detect where small mammals or possible predators might be. So the birds can actually avoid these areas and avoid being eaten. And therefore, they're going to be more likely to survive, more likely to reproduce and pass on this beneficial allele for detecting the UV light to their offspring. A new dog food has been developed that does not contain meat from cows, sheep, or chickens. The new dog food contains insects. The insects in the dog food factory are fed on waste vegetables. Sketch the pyramid of biomass for the food chain that produces food for dogs from insects. Label the pyramid. All right, so we need to put our producers on the bottom, which are the waste vegetables, and then the bars need to be the same depth and in the middle as much as possible. You will be able to do this better than I can because you'll have a nice ruler. So we've got the waste vegetables, and then the insects feed on the waste vegetables. So then the next one's up. There'll be less biomass in those insects than there are in the waste vegetables. And then the dogs will be feeding off of those insects in the dog food. They will be less again. So your pyramid should look like this. Describe 2 reasons why the biomass of the insects eaten by the dogs does not all become the biomass of the dog. Not all of the insect can be digested and absorbed by the dog. The dog also loses some of that biomass when it releases carbon dioxide from respiration. Explain how making dog food from insects can improve human food security in the future. So this means not running out of food. Well, because cows are no longer used in the making of the dog food, it could mean that there'll be more cows available for human consumption. Or if there are fewer cows being farmed or reared, it means there's going to be more space to grow plant crops to feed humans. Fewer cows will also mean that less methane will be released, so there'll be less global warming. Why is that relevant to this question? Because there'll be less climate change that could potentially damage our crops and food sources. Perhaps we can develop more foods for humans using insects. Number 8, almost there. Figure 8 shows when 2 mammals existed in Asia. Determine the number of years both Andrewsarchus and Brontotherium existed together. So if we look at the scale of where those bars are overlapping, we've got 38.5 million years ago to 36 million years ago. So they're overlapping there. So we just have to minus 36 million from 38.5 million, and that is 2.5 million years where they existed together. The oldest fossils of human ancestors found in this area are 700,000 years old. Andrewsarchus was a carnivore and Brontotherium was a herbivore. Suggests how the extinction of the Andrewsarchus could have resulted in the extinction of Brontotherium. I think there are different ways you can answer this question, but the way that I would go would be that Andrewsarchus may have preyed upon the Brontotherium. So no more Andrewsarchus would mean that the Brontotherium population would increase. And this increase in Brontotherium could have caused their food source to decline because that's all they've got to eat, and there's too many of them now, so they're all eating the same stuff. And with little food available due to the competition between the Brontotherium means that they gradually would starve to death. Information about extinct animals is often not clear because the fossil record is incomplete. Give 3 reasons why the fossil record is not clear for older species. Well, fossils are buried deeper, so they're harder to find. They are more likely to have been destroyed by geological activities such as earthquakes. Older organisms that were soft-bodied will have decayed and leave behind no fossils. Figure 9 shows the percentage survival of large mammal species in 4 areas of the world. The time at which humans first appeared in each of the 4 areas is also shown. So that is the black arrow, shows you where those humans appeared. And we've got a time scale along the bottom and our 4 different areas. A mass extinction is a rapid decrease in biodiversity on Earth. A student stated, "The data in figure 9 shows that humans caused mass extinctions." Evaluate the student's statement. When we have to evaluate, we must say how their statement is supported and how it is not supported. So there's definitely a decrease in large mammals after humans have entered the areas. The decreases occur at different times in each area, so this decrease can't be due to a catastrophic event such as meteor impact. The decrease is not always immediate, so that gives time for the human population to increase in their size before causing that decrease in the mammal numbers. However, this data actually only shows a decrease in the number of large mammals and doesn't show any other species. The decrease in mammal numbers in Africa is quite small if you compared it to Australia, for example, and the decrease is quite slow in Madagascar. The data only shows 4 areas of the world. There could be other factors causing the decrease, such as specific predators or pathogens, or perhaps there are abiotic factors that have led to the decrease, such as a change in climate. Even though there looks like there is a correlation between human arrival and the decrease in mammals, it does not mean that humans are the cause of the decrease. Correlation is not causation. And finally, give one disadvantage and one advantage of mass extinction events. Answer in terms of evolution. Disadvantage, you could have a loss of a range of alleles, so the biodiversity of the species. Advantage could enable the evolution of a new species. And that's it. Well done for getting through the whole paper. Good luck in your exams and see you in the next video.